do its thing. So Sarah, just tell us whilst I can see my bank of lights. We're just waiting for Facebook to kick in. Sarah, you work for the fantastic organisation NIAB. What's the connection with CUPGRA, with NIAB and CUPGRA, please?
Yeah, it's quite a long-standing connection. So, over 30 years ago, before the lab group was associated with NIAB, Eric Allen had an industry event where he was talking to some farmers about the research he was doing, and apparently some man from the Ministry of Agriculture at the time was like, "Why are we wasting our money on this kind of stuff?" And a lot of people were outraged and were like,
Wow.
"We need to make sure that we keep funding this kind of work. We keep funding work that's happening on farms for farmers, that's going to be relevant and able to be applied." And then when Eric Allen's lab group moved from the University of Aberystwyth to Cambridge, and then that lab group joined NIAB in later years, the association with CUPGRA kept going. So we've kept on having that really close relationship, doing high quality in-field agronomy research for farmers and other people in the industry. And CUPGRA keeps the work that I do grounded. So I'm very thankful for this close association. And it means that the work that I do actually gets out into the farming world and can make a difference.
Well, you stole my question, but let me ask it anyway, just to reinforce that. Does the relationship between NIAB and CUPGRA and NIAB, does that relationship work?
Yeah. It is super valuable to be able to say when I'm applying for grant funding that there are real farmers who care about the project that I am about to ask you for money for when I'm asking the government for money. So we have lots of letters of support from CUPGRA saying how a particular project will benefit the industry. And because the government, they don't know, they don't really understand what the issues of the day are. So it's really helpful to them to know that the industry actually care about the science that we're proposing. And then the other way around, I get lots of feedback from CUPGRA members on the research that we're doing, helping us to shape it better. And they also participate in the research. So we've got projects going on different on farms of different CUPGRA members, which is a really, really valuable relationship because it's all very well me doing trials on the trial grounds at NIAB, but there are times when it's like we need to have different environments, different conditions, and it would take me a lot longer to drive around and find people if I didn't have that network that I get through CUPGRA. So I'm very fortunate as a researcher.
Well done. And talking of fortunate or mercurial, I think this relationship that you have created with the CUPGRA Student Network is sublime, and that's the purpose of this broadcast today. All the social media is now live. My name is MacGillivray from Beanstalk.Global. This is Dr. Sarah Roberts from NIAB. So just to give you the titles, and especially for those people listening on the podcast, so CUPGRA, pre-event broadcast investigating cutting-edge potato research that's been carried out across the UK by CUPGRA Student Network. And if it's okay, Sarah, I just want to give a quick background to the CUPGRA organisation for people within the UK that may not be fully aware. And also we have a lot of international contacts that dial in. So CUPGRA, the Cambridge University Potato Growers Research Association, CUPGRA for short, is a charitable association of potato growers, agronomists, packers, processors, and other stakeholders in the supply chain who value independent, high-quality research. Based in the UK, it promotes the interchange of scientific research and field practice for innovative, sound commercial production of potatoes through its association with crop research organisation NIAB that Sarah was just talking about. This close involvement with NIAB researchers and specialists enables CUPGRA members to rapidly transfer new knowledge into practices for commercial advantage. Their key events is the annual Cambridge Potato Conference, which has been held this year on the 10th and 11th of December at Robertson College in Cambridge. And if you look at the post, you'll see the links to it. And today's broadcast is to give a quick taste of the cutting-edge potato research being carried out across the UK. We are very lucky to have 10 members of the CUPGRA Student Network who will be presenting at the CUPGRA conference in December, here to introduce their work and intrigue you. Each student is going to present a taste of their work in a flash talk, and we're going to learn so much for these intellectual powerhouses today, and who are also going to be key for shaping the sector's future in the UK and internationally. So Sarah, what should we do? Should we just get everyone in first and just do a whiz pop bang hello everyone? Everyone turn your video on, please. So we got everyone in now. And one of our cohort, Anika, couldn't make it today, but we got a pre-record with her a little bit later. So Sarah, what do you reckon is going to be the best way to get the best from our CUPGRA students? Shall we split them into groups and then roll through the little flash talks and then have a catch up at the end with everyone? Would that be the best thing to do?
Excellent idea, Matt. So we'll start off with 3 students who are working on stress-related projects, but I'm not going to tell you any more. I will hand over to Em, who will get the ball rolling.
No problem. Everyone else, can you turn off your videos, please? Well done, everyone. They get penalised. What a word! They get penalised if they're last in. Oh, my video's still on, so I get penalised. Right, over to you, Em.
Okay. Fabulous. Have we got my slides up? There we are.
Well done, Sarah. Thank you.
So, next slide, please. Fabulous. So, I'm Em. I'm at the University of Oxford, and I'm specifically studying the effect of waterlogging induced low oxygen in potato. So, my project has 3 axes. Firstly, I'm doing some whole plant phenotyping, looking at the effects of waterlogging on tubers, and then I'm using molecular biology as well to, on the one hand, look at the enzymes that we know sense low oxygen in plants and the gene expression response that is caused by waterlogging. Next slide, please. So, just to give you a little taster of what I do, these are my phenotyping results. So these are Desiree, harvested at 9.5 weeks, and they were flooded for 48 hours at tuber initiation. So we've got controls on the top and treated on the bottom. And if we start on the left, which are plants grown in stable conditions around 20 degrees, you can see that even that short 48-hour waterlogging treatment has quite a profound effect on tuber development. So you can see we've lost the mother tuber, it's rotted in the waterlogging treatment, and the treatment has caused this proliferation of lots and lots of tiny tubers, contrary to what we see in the controls. Now, we had some issues with our greenhouse temperature control this year, which has had not great effects replicability. What it has given me is the ability to look at the intersection between temperature stress and waterlogging. So if we go to the middle pane, we've got a cold treatment. So that's around 15 degrees. Temperature's getting steadily colder as the season progressed. And you can see that we've again lost that mother tuber and got lots and lots of tiny tubers. And you can see, if you compare with the first column, that the cold stress is already causing shrinkage, and the waterlogging treatment, the cold, meaning the waterlogging stays around for longer, is exacerbating that change. Now, if we switch to heat stress, which was spikes of up to 35 degrees during the day, but stable night temperatures, you can see that the mother tubers have actually made it through just fine, but that proliferation of tiny tubers is present in the control as well. So it is a response that we can see under multiple stresses. It's not unique to waterlogging stress. What we did see is that if you look at the waterlogging treated, so bottom right-hand corner, the skin integrity is a little bit less with waterlogging, but if you just look at yield, sometimes the waterlogged ones can actually come through better. So many things that interact in this treatment, and that, you know, sometimes waterlogging in the non-stressed or the cold stress is true waterlogging, but in the heat stress, that same treatment was actually experienced by the plant as a really good drink in a hot and dry environment.
Well done. That's you've obviously put a lot a lot of time and energy into it. I can't wait for the actual conference in we're just going to find my in December, so we could find out even more with you face to face. So Em, is that your slides done? Just so I know.
That is
Brilliant. Well done. So Sarah, could you stop the screen share? And we're going to move on to thank you. Could we move on to Favour, please, now? Here comes Favour. She's just turning on her screen. I'll just fill in whilst you're here. No, Favour's here. Okay, I'll go away. Well done, Em. This is and well done, Sarah. This is working very well. So Favour, keep your screen on, please. And if you say to Sarah as to what slides you'd like on.
Yeah, my first slide, please.
There you go.
Well done. Off you go, Favour.
My name is Favour, and I'm working on anti-transpirant to mitigate drought stress in potatoes. Next slide. So this is an overview of my research work on drought stress. And the first picture we have there is the experimental setup and the polytunnel, where we use 4 potato varieties, 2 drought level and 2 anti-transpirants to mitigate drought stress. The next is the graph showing the volumetric water content. At the volumetric water level, the irrigated plants were actually at 45%, whereby the drought stress were reduced to 30% of what the irrigated plant enjoyed. And the arrow there shows the period when anti-transpirants were applied at tuber initiation. The blue line is the irrigated plant, where the other lines are the drought plant. Next, we see the effect on plant water status. That plant there shows a wheated plant with drought plant without anti-transpirant after 21 days of stress. And the other one is also a drought stress plant, but with the application of VG, which shows that water content was high in that plant. We also took the measurement of stomata conductance using porometer. This also is to tell us how well the stomata is opened or closed with the treatment applied. And as we could see, the irrigated plant shows high level, compared to the drought plants. But among the drought plants, the application of VG and ABA, which are the anti-transpirants being used, as a higher stomata conductance compared to the drought level. Then we analyze for yield, and as expected, the yield for irrigated plants were high compared to the drought plants, but improvement with anti-transpirants were not statistically significant. Then for the last aspect of the research is the physiological disorder assessment, which is the most interesting part of this research. And with the disorders that were analyzed, among all of them, 3 stood out: scaly skin, internal brown spot, and jelly-head rot. Among the irrigated plants, they were low with these disorders, but with drought plants, they're high with these disorders. And with the application of VaporGuard and ABA, which are the anti-transpirants, we saw a drastic reduction, both pre-storage and after storage. Thank you.
Favour, well done. I can see you put so much work and effort into that. It'd be interesting to see the questions that we're going to get at the end of this segment for stress. Edwin, over to you. Favour, thank you. Could you turn off your video? Let's get Edwin in. And Sarah, if you could start Edwin's screen share for his slides, please.
Yep, let's get
oh, Edwin, just turn on your microphone. I think it wasn't on just then.
Yes, okay.
Yeah, we can hear you. Right, off you go, sir.
Thank you, Max and Sarah, and greetings to everyone across the world. And my name is Edwin. So thank you for joining the broadcast. So what you're seeing on the screen right now, it's more or less what Favour was talking earlier on. These are called the physiological disorders. So the unfortunate part of the physiological disorders is that we can only notice them when we want to utilize the tuber, and it's resulting in a lot of food waste and loss of money. So the reason why I'm talking to you right now is trying to reduce the number of tubers that ends up on a table looking like this. So if you go to the next slide, you can see that probably the biggest question you want to ask is, why are we having these kind of physiological disorders? So in a broad spectrum, I think the main answer will be the climate change. The impacts of climate change, we're experiencing increased temperatures, drought, resulting in water stresses. So these water stresses are the major causes of these physiological disorders. But the main thing with these physiological disorders is that they develop, maybe we can notice them at harvest. They develop very slowly. Sometimes they can even develop 3 months after harvest whilst the tubers are in store. But currently, despite the current efforts where farmers, agronomists use some phenotyping methods, but these current methods are very destructive, they're time-consuming, labor-intensive, that we cannot afford during these times. So my possible solutions, which I'm suggesting, I'm still in the early stages of my research, but we're trying to replace these current methods with sensing technologies. Imagine monitoring your crops whilst remotely, and this can save us a lot of time. It doesn't need so much labor, and it's not destructive as well. So for example, if you're using a thermal imaging, which can tell you that this crop is under certain temperature, the leaf temperature, the canopy temperature, that is linked to the water stress, and eventually then linked to the occurrence of physiological disorders at a later stage. So this is a game changer, using the remote sensing in so many ways. For example, if you look for farmers, growers, agronomists, it's saving them. This will save them a lot of money in terms of investment, in terms of losing so many tubers. Try to think of it. You get a tuber that is exposed to water stress and then develops a disorder 9 months, and you after harvest in the store, and we spend the whole 9 months of investment on a tuber that is going to develop a physiological disorder. So it's a game changer if we are able to know that this tuber or these patches of tuber are going to develop disorders maybe 3 months later after harvest, so that we can make an informed decision to distribute these tubers before they develop these disorders. And despite that, we're going to touch also the touch base of it's not just the economics. You're going to touch base the aspect of environment, because right now there's COP29 going on, which states that 33% of the food we produce, it goes to the food waste. So as a sector, as industries, potato sector, if we manage to reduce the amount of food we waste by 50%, we would have done so much impact to the environment, economic, by producing responsibly. So thank you, and I hope this cutting edge will be very effective in the future.
Edwin, well done. I can congratulate you. I was just trying to segue this. I was filming in a greenhouse recently. Sarah, could you stop the screen share? And and Favour and Sarah, can you all come in? Edwin, I was filming in a greenhouse recently, and the owner of the greenhouse was stating that there's so much energy going into R&D of harvesting of tomato crop, when actually the energy for R&D needs to go into agronomy, pest identification, and disease identification. And you've just intimated that within the potato sector, because especially with the food waste element, we always talk about food waste. 1.2 million bananas are thrown away in the UK every day, and so. But actually, if we can make the crops more efficient in the ground and ready for harvesting, there's so much profitability held up within the supply chain that your research is as you've intimated, will be a game changer.
That definitely will be a game changer, because if you manage to monitor crops whilst in the field, not just monitor it, but in a cost-effective way, I think we save so much. You can have a farmer producing at a very better extent than the current one.
Yeah, and I'm sure you know, and we'll talk about it at conference, but the potential waste levels that happen in the field because of the lack of identification for what you've just. I bet it is. I bet it is significant. So again, to have your R&D coming in will be a game changer. So back to the group as a whole. Stress. Sarah, have we got any questions for the group?
Haven't seen any coming in from the audience, but I've got a couple to kick us off.
Go.
Can I start with you?
Sure.
So you said you impose the stress at tuber initiation, I think. What do you know what would happen if the plants experience waterlogging either earlier or later in their development, or can you have a sensible guess for us?
Yeah, so as a bioscientist, first what we wanted to do was to gather a kind of reference data set that we can later compare back to with modified potatoes. And we chose flooding at tuber initiation because the very little data there was showed that would have the biggest effect on tuber development. So we would tend to, if we flood a commercial variety earlier, we will tend to see less damage because the root system is actually relatively resilient. What you might end up there is problems towards the end of the season. If you've got a set harvest date that you can't push back, you might end up with same problems. However, if you're able to leave the crop in the ground for an extra couple of weeks, you might be able to balance that out. Where we come in with kind of later tend to see more kind of rotting and tuber. You'll see with my plants, we kind of see a range of tuber sizes, and you may not see that. If you get later, you may see your remaining large tubers, which are more liable to develop defects, and then a range of very small tubers that are never able to catch up and are essentially then becoming either waste or secondary production that won't hit the market for human consumption.
Yeah, not the valuable stuff. So you just halted that growth. Cool, thanks. Favour, I've got one for you. Does the effectiveness of your anti-transparents vary with when you apply them? And do the plants have to be like unstressed before they get applied, or do you apply them once they've gotten stressed?
Thank you for your question. So anti-transparent, we've used it in the crop group here at Anika Damm with other crops, but this is the first time we're using it with potatoes. And with the research before, it has shown that anti-transparent does not really work with irrigated plants. It's effective when it's drought. So we allow it to drought for like 2 weeks, then we set in the treatment, applying anti-transparent, then we start to study the effect. That is how it works.
Yeah, so that's super practical because it means that you don't have to know that the drought, you don't have to prep for the drought to come. You can wait until it's set in and then apply the anti-transparents to then protect the crop.
Yes, we actually look at because with anti-transparent, the stomata is blocked. And you know, with stomata, it's work with photosynthesis. If it's blocked, then photosynthesis will not take place. So we actually target a certain stage before it's applied. So we target the tuber initiation stage. So that is when we apply the anti-transparents. Then, because at that stage, drought also, we set in drought, then we put it on.
Yeah, great. Thanks. And Edwin, of the different methods that you've been using to understand, like to record the potato stress, are they all telling the same story, or do some of them associated with particular kinds of physiological disorders?
Okay, thanks, Sarah. I think in most cases, the most commonly used one is the thermal imaging, where you detect the crop temperature. But it's because I'm still in the early stages, but the preliminary results are showing that it's also dependent on the variety which you're using, the crop response to drought. Well, I'm not getting much from the chlorophyll fluorescence, so I'm tempted to drop it off, and then use more of hyperspectral and thermal imaging. But the most promising one is the thermal imaging, where it takes the temperature of the crop.
Okay, cool. That's really useful that you're narrowing down the different monitoring options, because sometimes there can definitely be too many choices for growers to pick between technologies. Got an eye on the time, Max. Shall we head on to this?
Well done. Team Favour, Edwin, thank you very much. If you could turn off your videos, and let's bring in Fabian and Brandon for tuber quality and storage. Sarah, this is going very well. I think we're like the one show dream team for cutting edge potato research. What do you think? Do you think we'll get signed up by the BBC?
Yeah, I reckon so.
Yeah,
I reckon they'll be phoning us up shortly.
But seriously, just we're only 24 minutes in, and you must be so proud of the students. You could just see that level of dedication and intellect that they've got, and to be involved with assisting them to take the sector to the next level. It must be very enthralling.
Yes, no, it's a privilege to see all the different kinds of research that are going on, and to be able to bring people together to share it, and also to make that connection with industry and make sure that the research is getting out there. Should we hand on over to Fabian?
Well done. I was going to say let's bring Fabian in. Fabian, where are you? Turn on your video and your mic, please.
Hello.
Well done. Now, just to say, we were in the green room. Fabian comes from the coolest country in the world, Colombia. But I'll be quiet and let him get on with what he's going to tell us. Go, Fabian.
Thank you so much, Max. Thank you so much, Sarah. Thank you so much, everyone, for giving us the opportunity to be here. My name is Fabian Villamil. I'm from Colombia. Thank you, Max. My project is about understanding potato endodormancy to reduce mainly the food waste. Next slide, please. So, because the point is here in the UK, we mainly have just one harvest season. So we need to produce the potato tubers, and we need to store these tubers for up to 9 or 10 months to ensure 1 year availability. However, during the storage of these potatoes, the sprouting causes food waste, economical losses, and this finally affects the processing industry. There are lots of different alternatives to extend the potato storability, like the use of the CIPC, the use of controlling the environmental conditions in which we store the tubers. But there are many different problems associated with these alternatives. For example, the CIPC, which was the principal chemical used to extend the potato storability, was banned from the UK and the European Union because they're harmful to the health of the human. And as well, the use of the low temperature increases the reducing sugars, which affect the quality of the potato tubers by the processing industry mainly. So the aim of my PhD is to understand what is the effect of the pre-harvest factors, those factors that we can control mainly on the field. What is the effect of these factors on the potato dormancy extension? But the point is, this is a really complex trait, is controlled by many different factors, and there is not much information about this. We found a research gap in this information. So we have narrowed a little bit the project because we are mainly focusing in the effect of 2 of these different pre-harvest factors, such as the nitrogen, which we applied on the field while the plant is growing, and as well the genetic background. So the idea is to bring together all of the results for these different trials that we are performing, and something similar what Edwin mentioned, to create different predictive models in which we can predict the time in which, for example, a determinate variety or cultivar will start to break the dormancy. For example, imagine that you have all of the tubers in a really big pond, and with camera you can easily detect and quickly detect when they are going to break the dormancy. So it's as well to reducing the food waste, and as well to reducing the economical input for example, with the nitrogen on the field. We found for some trial that we have performed that the less nitrogen we applied, the longer the potato tubers will be stored. So it's about food waste and as well to reduce the economical losses, to reduce the effect of the fertilization and well, many different problems. So thank you so much. In the next slide, you can find some details about me, but well, thank you so much.
Well done, Fabian. Again, it's your research. You can see when you deploy it, it's going to be groundbreaking. Fabian, thank you. Brandon, over to yourself. Fabian, if you could turn off your video, and Brandon, come on in.
Hello.
Hey, Brandon, just to check, have you had a refill of your drink? As we're talking in the green room. Brilliant. He's all set.
I mean, yep. It's all blurred out though because of the filter.
No problem. Off you go. Off you go.
No worries. So coming on to my project, I've literally only just embarked on this in the past month and a half. But I'm looking into bruise resilience within Morris Piper potatoes, given that it's one of the most commercially grown cultivars in the UK, and also accounts for 4% of the global food crop. Potatoes generally, not Morris Piper. However, I can talk to you about what I'm planning to do. So my research will focus around using RNA sequencing to isolate candidate genes for bruising. And this will come from two types of bruising that I'll be doing, which is pressure-related bruising and impact-related bruising. And then from those candidate genes, I'll be selecting about 4 genes, which I'll then be looking to using CRISPR, potentially knock out those genes in order to create several new lineages. And then that way, for each gene, I'll be able to produce modified or edited microtubers. And then from that, grow them into full crops, which I'll then be able to do a comparison of between the unmodified parent and the gene-edited tubers. Now, one thing to take into account with this is the 2 types of bruising, the different stages of bruising. So I'll have to be taking samples from different time periods after the initial bruising has taken place. And also is kind of the sourcing of the actual potatoes themselves, because if you were to just get them off a supermarket shelf, for example, you don't necessarily know the background of where they've come from. In some instances, it's been documented where potatoes have been held in long storage and then they've been taken out only to, no matter how carefully you handle them, instantly bruise, even just by holding them. So when it comes to things like that, that's what I've got to consider with regards to the origin of samples that I'll be working with. But following that, I'll be then conducting a comparison between the bruised tissue and the original parent tissue, including physiological traits. So I'll be taking into account things like phenolic content. And then, provided that everything goes as planned, which, as I've heard about PhDs, isn't necessarily the case most of the time, I'll then be evaluating wider characteristics focused towards an industry context. And with that comes the field and storage trial side of things and analyzing consumer needs. But yes, so that is the current stage of my research. However, I'm not even 2 months into it, so I will try and answer any questions as best I can.
Brandon, well done. It's the only thing you can ever predict is change, isn't it? Sarah, could you just stop the screen share? And Fabian, Sarah, can you come in? Brandon, the only thing you can predict is change. And I suppose if you set off in one particular direction with your research, you've got to have that flex, haven't you? Knowing that it's going to do this.
Exactly. It's one of those things. No matter, whenever I've spoken to anyone about a PhD, they've always said that something will always go wrong, and you need to account for it. You need to always account for that.
Sarah's nodding. Fabian's smiling. Sorry, Brandon.
Yeah. No, it's fine. So, like I said, with that, currently I do have those 4 objectives, but I need to kind of leave some leeway. So the 4th objective is very much if by some miracle things were all done to plan, and even finished early for whatever reason, then I can start looking into that kind of wider context.
Yeah. So Brandon, here's the big question. What does success look like for you on this PhD? What does success look like?
In an ideal world, I'll have miraculously unlocked the secret behind potato bruising, which I believe has stumped many a researcher over the decades. In reality, I'll probably end up, well, I hopefully will end up finding at least one gene, ideally maybe one or two, that are directly correlated to the bruising, and by knocking or potentially knocking these genes out, that can possibly reduce the bruising, but not in a colorimetric way. The colorimetric analysis has already been done by other research. I'll be looking more towards the mechanical effects on the cell, so the starch production, the rupturing of cell wall, that kind of thing.
Wow, well done, well done. Fingers crossed. If anyone can do it, you can, Brandon. Sarah, have you got a question for Fabian? Just looking at the time, and we'll use that, Brandon, if that's all right. We'll allocate that as your question.
Not a problem.
Yeah, Fabian. Of the pre-harvest factors that you've considered so far, which one of them has had the greatest effect on dormancy duration?
Well, I think that mainly the cultivar, the variety has a really big impact. The genetics behind is really big impact. It has a really big impact because there are some potato cultivars which will break dormancy really, really early, and there are some other which can break dormancy really, really late. As part of my PhD, we are like looking for particular genes which are involved in this, but it's quite complex because there are lots of different genes that has impact this. On the other hand, nitrogen as well can have an impact, but I think that it's quite difficult to measure the nitrogen by itself because it can be affected as well by the rain, by the different environmental conditions that can affect the potato on the field. So it's quite hard to understand the role of nitrogen by itself. I think that maybe the big one is it could be the genetics behind the trait.
So the most practical thing for farms at the moment is picking that variety. Yeah, thanks, Fabian.
Well done, Fabian. Just on the Q&A, on the box on the bottom line, Mark has got a question for you. If you could answer him online, and Mark wants to catch up with you as well offline or at conference. If and Fabian, if you could just look at that question from Mark, that'd be great. Fabian, if you could turn off your video, and Brandon, if you could turn off your video, thank you very much. And if we could bring in for the IPM section, John and Vongai, that'd be great. As we wait for them to come in, Sarah, how do you source the students?
Oh, it's a word of mouth thing at the moment. So if anyone watching knows someone studying for a PhD on a potato related project, send them our way.
Wow.
There's a sign up form on the CUPGRA website, which is CUPGRA.com, and you can join the student network like that. Yeah, brilliant. Word of mouth.
Well done. John, turn on your video. We want to check that you're ice skating up north in the room. John was saying that he couldn't get out of his front door because of the 12-foot snowdrifts. So John, just turn on your video. We can hear you.
I have actually turned my video on, but for some reason it's not.
It's on. There we go. Okay, I will turn off my video. John, over to you, sir.
Right. My project is looking at developing a push-pull system for the control of aphid vectors of potato viruses. So what's push-pull? Basically, it's a pest management strategy that's been trialled and quite successfully in a range of pest situations, so veterinary, medical, and agricultural crop pests. It's a technique that relies on using the stimuli that the pest uses to locate its host. And it confuses it by firstly either masking or repelling it. That's the push side of it, and simultaneously you provide it with an attractive stimuli to move it to another place where it's either not going to cause any harm or you can control it a little bit more easily. Key to the success is actually identifying the most effective stimulus, and for aphids it tends to be vision, because aphids tend to locate new crops and hosts by vision, and what they tend to look for is a green plant against a dark background, the contrast, which is why aphids, when they arrive in a crop, tend to arrive at field edges in farm gateways, and it's one of the things that make row crops like potatoes particularly vulnerable to aphid infestation. There have been sort of various attempts to actually mask the potato crop from there. So some of the growers may be familiar with straw mulching, which has proved effective. But I was intrigued by, if you can show me my next slide, a study that was done by some sugar beet growers. If I could have my next slide, please. There we go. Yeah, this is a trial that one of my supervisors showed me fairly early on in my project. What they're doing there, they're actually using coloured dyes to actually try and hide the crop from the aphids. So red, most insects don't actually see red, so that's the idea of masking it. The technique has actually been trialled in some other crops. I'm aware of it being trialled in carrots, and I know some growers are actually trying it this year in potatoes. So that'll give you a mask, which is your kind of push. But it got me thinking the gap in the situation is you actually need a pull crop, so you can actually hide the potato crop and actually pull the aphids somewhere else. So the early stages of my project, I'm looking at developing a visually attractive pull crop. So the first challenge I actually had is how can I successfully colour plants? So if I can have my next slide, please. After a bit of trial and error with food dyes and all sorts of weird and wonderful things, we came across the idea that artist acrylic paint applied through an airbrush is actually a really effective way of actually colouring plants. So you can see on the left there's some barley plants that I've sprayed red. The next stage has been basically to see, well, I need to test whether the aphids are responding to the colour and not the paint. So I've been running a series of experiments to actually see if I can actually if the paint actually puts the aphid off feeding. And as you can see from the picture on the right, they're perfectly happy with the acrylic paint. So I've been testing that statistically, and I'm just running the last few experiments through on that. But thus far, it looks like acrylic paint is a successful colour that I can actually now go on to use, and I'm just about to progress onto some landing studies, which will be commencing next month.
John, well done. Pull crop, that could be, well, it's going to be revolutionary, isn't it?
Yeah, well, if I can get it to work, it's got applications for sugar beets, a lot of sort of high value horticultural crops.
John, well done. Thank you. John, if you could turn off your video. Vongai, if you could turn on your video, please. I'll just turn on my video whilst we wait for...
I'll move on to... Yep, here's your first slide.
Okay. Vongai, are you there?
Yeah.
You're here. Well done. It's not you. It's Zoom, because it's coming all the way over from America. Over to you.
Thank you very much, Max. Hello, everyone. My name is Vongai. I'm here to talk to you about a project where we are trying to use cover crops to manage root-lesion nematodes, that is the Prattlinger species. So this nematode is quite important in potatoes, cereals, and other UK commonly grown crops. And you really don't want much of this nematode because in potatoes, as few as 2 nematodes per gram of soil can cause significant damage. And we also have the potato early dying, which is a complex between root lesion nematode Prattlinger penetrans and verticillium wilt, which causes further damage. At the moment, we have limited nematicides that are approved for use in the UK, and rotations are not feasible because of the polyphagous nature of Prattlingers. So what we did for the past 2 years, we did experiments, field experiments, to try and test different cover crops in fields and see their potential on reducing Prattlinger species at the same time without negative effects on beneficial non-target nematodes. Next slide, please. So the graphs I'm showing you on the left is the root lesion nematodes, the Prattlingers, which is the bad nematode. On the right, we have the beneficial nematodes. So what we did, we planted cover crops between June and September for a minimum of 3 months. Then we assessed nematode populations at planting, then 6 weeks after planting, and 6 weeks post incorporation of the cover crops. And from the cover crops we tested, this is just data for one site, we can see from the highlighted circle areas on the left, we have French marigold and oilseed radish, which managed to reduce Prattlinger species But we also have Indian mustard, Brassica juncea, which actually increase the nematodes, meaning it's not a good cover crop to use for managing Prattlinger species. And on the right, that's the assessment of the beneficial nematodes. And you can see that all of the tested cover crops actually increase the beneficial nematodes, which is a good thing because beneficial nematodes make up 80% of all the nematodes in the soil, and they actually improve soil nutrient cycling. And thank you very much, and see you at CUPGRA if you're coming.
Well done, Vongai. Sarah, if you could stop the screen share. Sarah, if you could come in. John, if you could come in. And Sarah, if you got any questions for them both, please.
Yes, I do. John, I'll start with you.
Hiya.
Can you describe what it might look like on a farm which is able to implement the push-pull system? Like, is this happening within a field, between fields? What's the scale?
It will be. I mean, if I can get it to work, the sort of the concept will be that there'll be a coloured patch in an adjacent field, or possibly 2 or 3 fields away, because there is some evidence that shows that aphids look for. Excuse me. You get more aphids in a row in an environment dominated by row
Sarah,
crops.
if you got any questions?
Sorry.
Keep going, John. We just had some
Sorry,
feedback. Keep going.
Yeah, so I think the problem with aphids is that they've got very good control of their flight over when it's all calm, but obviously you don't get calm very often. And so there's an element of stochasticity or randomness in their flight. So my idea is if you can actually pull them, have them aim for something well away from the potato crop. So yeah, I imagine you've got a better chance of actually pulling them there, and if you then combine it with the masking from the sort of either by a dye or a straw mulch in the potato field itself.
Yeah, and a super quick one. Is there any worry about acrylic paint and microplastics getting into the environment and the water?
Well, at the moment the acrylic paint is just there so I can actually prove concept.
Okay, excellent.
So yeah, if I can demonstrate it'll work, then there's then the next stage is actually to find a suitable thing to use in a commercial situation. But at this stage, it's proof of concept at first.
Yeah, as so much research is.
Yeah.
Thanks, John. Vongai, was there any variability of the effect of cover crops between soil types, or were your experiments just in one location? Oh, you're still muted.
Vongai, just turn your ears on. So, well done.
Thank you very much, Sarah. So we did experiments in 3 sites, 2 in Scotland and 1 in the Isles of Scilly. And we did see different performance of oilseed radish in particular. In Scotland, we did see the nematodes going down, but it wasn't statistically significant. But in the Isles of Scilly, the one that I just presented, you can actually see that it was significant. So I think the performance of the cover crops do vary with the soil type.
Yeah, or potentially maybe weather as well. Should we keep moving
well
on?
Done. John, Vongai,
Thanks,
thank you very much. If you turn your videos off. And Francis, Ronald, if you could turn your videos on. Sarah, just a quick question. Do you do much collaboration with industry? I'm learning so much today, and you can see how this R&D that everyone is doing is going to be beneficial to the sector. Do you get businesses looking to collaborate with yourself and the students?
We haven't had any sort of student network industry collaborations set up yet. Although I think I'm right in saying that a number of our different students are connected with industries sort of as part of their project, so not through the network.
Okay. But would that be beneficial through CUPGRA, through this broadcast, if we could have companies that could see that if they assisted the likes of John or Vongai to assist them, it would be beneficial all round.
Yeah, okay. And having industry input helps shape projects to make sure that the outputs are relevant for real growers, real packers, real agronomists, etc.
Well done. Let's make that happen. Francis, turn your video on, please.
Yeah. Can you hear me?
We can. The floor is yours. The floor is yours, sir. Off you go.
Okay. Thank you, Max and thank you, Max and Sarah, for organizing this broadcast, and hello to everyone who is watching us wherever you are. My name is Francis Kawalya, and I'm from Harper Adams University in UK. And I'm going to talk briefly about my research that is looking at optimizing partial biofumigation for management of potato cyst nematodes. Next slide, please. So I'll start by giving a simple background about partial biofumigation. So this is simply a method that involves using brassica cover crops to suppress. It's just a method that involves using brassica cover crops, for example, oilseed radish, Indian mustard, rocket, and Ethiopian mustard, to suppress soil-borne pests and pathogens. For example, when you look at the previous speaker, Vongai, she was looking at one of the brassica cover crops that were oilseed radish against Platyrhynchus penetrans. But for my case, I'm looking at it from the angle of managing potato cyst nematodes because they are very tricky nematodes. They are one of the tricky nematodes to manage in the field. Then the mode of action behind the suppression of soil-borne pests and pathogens using brassica cover crops lies in the ability of the brassica cover crops to produce compounds called glucosinoids within their roots, and they release these compounds into the soil. And once these compounds are released into the soil, they are converted into different biocide organic compounds, for example, the isothiocyanate. And this conversion is done by an enzyme called myrosinase, which either comes from the tissues of the brassica cover crops and leach together with glucosinoids into the soil, or this enzyme can also be produced by some specific soil microbes that use glucosinoids as their source of energy. So in doing so, they convert the glucosinoids into the different biocide organic compounds that are responsible for separation of these. So we are building from that background to develop optimized partial biofumigation to manage potato cyst nematodes. And in this research, we are taking different approaches. And one approach that we have already worked on is the identification and selection of brassica cover crops that are more tailored to partial biofumigation. And specifically under this, I was looking mainly at the ability of the different brassica cover crops to reduce the viability of potato cyst nematodes or suppress potato cyst nematodes. And from the studies we have conducted so far, we have selected a few promising candidates. And then the next step that we are looking at the moment is to see if we can improve the selected brassica cover crops in terms of glucosinate production, because as I mentioned, it's the most important compound when it comes to biofumigation. And then also looking at if we can increase the root production, because increasing root production would increase the root surface area for the release of the glucosinates. So take it in this situation, you've increased the production of glucosinates in the roots, but now you need a larger surface area so that they can be released into the soil. Then the next approach we are looking at is to see if we can boost the activity of myrosinase enzyme in the soil. So what it means is like the plants are producing more glucosinates, but now you need higher levels of myrosinase or higher concentration of the myrosinase enzyme to convert it into the biocide organic compounds. So that at least at the end of the day, you have like this complete system, higher glucosinase, higher conversion into biocide organic compounds. But at the moment, everything here is being optimized under glasshouse conditions. But the plan is to test the optimized system under field situation, because that's where the potatoes are grown, and that's where you expect the potato cyst nematodes to be a problem. So in the nutshell, that's the simple overview of my project. And I hope to see most of you during the CUPGRA conference in December to discuss more about my results from some of the experimental work. Yeah.
Well done, Francis. That's very impressive. Very impressive. You can see how much work that you put into that. Thank you. Francis, if you could turn off your video, and Ronald, if you could come in, please.
Thank you, Max. Hello, everybody. So I'm Ronald, and I'm in my second year PhD at Harper Adams. And I'm looking at how we can use chitin to reduce potato cyst nematode populations, particularly in England and Wales. So if you move to the next slide. Potato cyst nematodes are a big problem within the United Kingdom. And at the moment, the management of the pest is really challenging because there is very stringent rules when it comes to the use of pesticides, and also some markets especially prefer specific varieties which have less tolerance to the pests. So at the moment, people are trying to find alternatives that are sort of environmentally friendly that could be used to manage this pest. And that's where I come in. I'm looking at the use of chitin in suppressing potato cyst nematodes. So chitin is sort of like a polysaccharide that is rich in nitrogen, and it is almost similar to cellulose. And because of its specific properties, has been found to be suppressive to nematodes. So as you can see here, these are some of the mechanisms that are sort of involved in the suppression of nematodes. So my project is basically looking at how we can optimize the application of chitin using field populations of cyst nematodes. And we also want to investigate the underlying mechanisms involved. For example, in my specific project so far, we've looked at the activity of certain enzymes, because when we add chitin in the soil, it promotes specific microbes which can produce enzymes, and these enzymes can target the nematodes, and this enhances their decline in the soil. So far we've done a preliminary greenhouse experiment, and we added chitin in the soil, and then we measured the viability of the eggs inside the potato cyst shell. And we could find that chitin and chitosan, which is also related to chitin, they kind of had an effect in terms of the suppression of the nematodes. So we could see that after 8 weeks, there was a slow decline in the viability of the nematodes. But over time, this rate of decline sort of was the same across treatments that even didn't have chitin. So in the next phase, we're trying to see how we can optimize this application rates of chitin so that we can enhance the efficacy of applying chitin basically in the soil. So this is just a disclaimer that just like any other kind of control method, it's not a panacea to the problem, but it can definitely be introduced in the IPM scheme. So yeah, thank you very much.
Ronald, well done. Again, brilliant. Hugely impressive, as are all of the others. Ronald, if you could just turn off your screen, and I'm going to do the screen share now. So Sarah, if you could turn off your screen and stop your screen sharing. Thank you, and wish me luck, everyone, because I never get these going very well. But here we go. This is just before I start. This is Anika, who couldn't make today, and you'll hear that I think she's going to be first on, but she's actually last on. So just excuse my little typo at the beginning. Here we go. Have you on? This is a very, very exciting broadcast that we're doing with you all on CUPGRA. We've got a lot of you to wrestle through. So let's start with yourself. Tell us about your slides, please. Tell us about your title of your slides.
So yeah, I'm Anika. I'm a PhD student at the University of Cambridge at the Crop Science Centre, and I'm working with plant parasitic nematodes. The title of my slides is going to be Disrupting the Regulators of Cyst Nematode Parasitism, and that's basically what my research revolves around. Should I start my slides?
Yes, please.
Okay. So as I told you, the title of my talk is Disrupting the Regulators of Nematode Parasitism. And as you can tell from the title, in my PhD, I work with plant parasitic nematodes. And given the audience, I'm sure you all know them as very damaging pests that cost agriculture a lot of money. And since they're so damaging, we try to find novel ways to control plant parasitic nematodes. And the approach I'm taking is I look a little bit at the nematode side, and I want to understand how the nematode regulates its parasitism to then block it. And the key to nematode parasitism are these small molecules termed effectors that the nematode secretes into the plant. And with these effectors, the nematode can manipulate the plant. For example, they help when the nematode has to invade the plant root. They can also suppress plant immunity, and they can actually manipulate the plant to make a feeding site from which the nematode then withdraws a lot of nutrients. So as you can tell, the nematode heavily relies on these effectors to infect the plant. And if we now think about a way to stop the nematode from doing that, we could try to target these effectors directly. That's an approach that's actually already happening in nature. So plants already have evolved some genes that can recognize these effectors and then are alerted, oops, there's a nematode around. But we don't want to just target one effector, and that's why we chose another approach and took a step back. And we actually want to find regulators inside the nematode that regulate the production of all of these effectors. And the advantage of targeting such a central regulator to nematode parasitism would be that by targeting this one gene, we could actually target a lot of effectors at the same time, and we can take away the key tools that the nematode needs to infect the plant. And we already found such a regulator that regulates the production of a lot of effectors. We can also show that by knocking down this one regulator, we can actually reduce the ability of the nematode to infect the plant by over 80%. And we set this up in a whole signaling cascade. It starts with the host signal and it ends with the nematode invading the host root. And now we want to translate this knowledge into a solution that can actually work in the field and that you guys in the farm could use. So yeah, that's a brief overview over my research.
Excellent. Okay, so just as I get the screen back on, just a quick question. How do you hope that your research will change UK farming practice in both the short and the long term, please?
So this is basically a novel approach to target nematodes and target nematode parasitism. So this gene isn't targeted by nature. It's not in contact with the plant. So we hope that this is a strategy that we can basically add to the current strategies that already exist. And it's probably a little bit more long-term focus. So now we are understanding the mechanism, and now we want to translate this into something that works. For example, one thing that could work is we could try to get a product that blocks this gene in the field that we could apply. We know it actually starts with the plant. So the plant produces some molecules that the nematode regulates, that the nematode recognizes, and then basically upregulates all of these processes that I just showed you. So we could actually try to breed for plants that do not activate nematode parasitism. And that is something that we could add to the genes that we already have, the resistance genes. So that would be basically a new kind of class for that. So basically, I hope to understand something fundamental that's happening in nature and then use this knowledge and basically produce new products or strategies that people could actually use in the field.
Excellent. And do you think that's is this going to work with both PC and species, do you think?
So hopefully. So we think that these molecules are probably conserved across host plants. So I don't know if both recognize the same signal in the plant, but surely they would recognize some signals. So we know that there's some diversity in the signals, and that's plant trait. So hopefully we could remove something in the plant that the nematode needs and then doesn't activate the parasitism. So I do not know yet the differences between the two species, Lavadera rostopiensis and Pallida, but I'm sure that we would find something that either both or one or the other responds to, and try to breed for that. But yeah, that needs more research and more knowledge.
Okay, I think if anyone's going to find out, Anika, you're going to find out. Anika, thank you very much. Thank you.
Well,
There we go, team. So let's just turn my video back on. Sarah, could you turn your video back on, please?
Of course.
So we're slightly overrunning. I put another pound in the slot to give us a couple more minutes. So this has been so intuitive is the wrong word. Insightful. I've learned so much. My head is all over gaggle. It's got me really excited about CUPGRA, because just to reiterate, we're going to have these 10 fantastic people on the second day. And Sarah and I are going to be, in effect, Sarah, repeating this exercise, but in person, so we can find out more and have even more interaction with the crowd. So Sarah, that's going to be very exciting, isn't it?
Oh yeah, it's going to be great. Do we have time to ask Francis and Ronald my questions, or are we running over too much?
No, bring them in. Let's get them in. Francis, Ronald, in you pop, please.
Francis, I'll start with you. Is there a minimum growing time for your cover crops in order for them to have an effect in the soil on the PCN?
Apparently, what's available. Yeah, hope I'm not muted.
No,
we can hear you. You're good.
Thank you for your question. Apparently, what's available, they have to grow between 12 to 14 weeks. But recently, I've been having a chat, and then we are trying to see if there are some specific brassica cover crops among those that I've selected that can grow past the 14 weeks, because if they can grow long on the land, that means the activity would be a bit, the efficiency of them producing these compounds would be a bit elevated. That means the activity in terms of separation would also increase. So that's what we are targeting and trying to see. But the minimum at the moment is between 12 to 14 weeks. But if you get plants that can grow after that, or if we can think of a way of prolonging these plants on the land, especially those that can that can withstand the winter when no crop can easily grow on the land, that would be great. So we are also trying to explore that possibility. It just came up in the research. It was not among the original objectives, but it just came up as I went on. So that's what happens with the PhD. A lot of things come on that way. Yeah.
It's so important to be responsive in a project. So well done.
Yeah, thank you.
Ronald, my question for you, is there, what source of chitin are you using? And is there any variation between chitin sources in the effectiveness against PCN?
Yes. So at the moment, we just worked with pure chitin, and it was sourced from shrimp shellfish. Yeah, but there are different sources. So chitin can be found from mushrooms, for example, the mushrooms that we eat, and then some black soldier fly, and then obviously the shellfish. And the different concentrations of chitin. So for example, usually shellfish has the highest concentration of about 25% of the shell waste, and about maybe mushroom and black soldier fly have around 12% of the chitin. In terms of, if I understood you correctly, do these different sources have different sort of controls, like efficacies towards PCN? Yeah. I have to investigate that further, but we know that so far chitin doesn't come on its own if it's a crude material. If it's maybe from mushroom, it comes with other compounds also. So it would be interesting maybe if those compounds also have effects on the nematodes. But this is something that obviously we're going to explore further, and maybe ask me again next year this time.
I will. Thank you.
Well done, Sarah. The witching hour is upon us. Why don't we do this? Sarah, I'm going to go for this up to close. Can everyone who's not got their video on, turn their video on, and then Sarah, wrap up for us. This has been such an impressive group. Go on, Sarah, wrap up for us for these lovely individuals.
I am so pleased for the short snapshots that you guys were all given our audience today, and everyone who will pick up the broadcast after it's been published. Thank you so much for presenting. Thank you very much for watching, everyone at home. And come and find out more. Come and talk to these bright young researchers as they work to improve potato agronomy and our understanding of potato production and just how potatoes work. Come and see them, come meet them, come ask your questions in person at the conference. which is in early December, and we'll have a link to it along with the broadcast, so you can book your tickets. Thank you very much.
So, Sarah, well done. And just to say to everyone, John, M, Ronaldo, Brandon, Edwin, Fabian, Vongai, and Francis, thank you very much. To me, you are the future of this particular sector. We need to support you, not only NIAB, but also the trade, the industry. It's interesting, some of the comments I'm getting in from WhatsApp from people in the trade who are watching this. So hopefully they'll come to CUPGRA and engage with you. We've got to collaborate more for the long-term benefit of the sector. And as I say, you are the future. So we look forward to seeing you at the CUPGRA conference in Robson College in the middle of December. Shall we try and do a collective thank you very much, good night, as it's now 9 minutes past 6. So here we go. After 3, we're going to go thank you very much, good night. 1, 2, 3. Thank you very much.
Good night.
Well done, everyone. We'll see you in December. Well done.
See you. Bye.